Good morning, ma'am. My name is Naimay Ali, BS Criminology. Today, I will be going to discuss and explain about the genetic basis of cancer. Let's get started. First, let's give the definition of cancer. Cancer refers to any one of a larger number of diseases characterized by the development of abnormal cells that divide uncontrollably and have the ability to infiltrate and destroy normal body tissues. Cancer often has the ability to spread throughout your body. 1 out of 2 men and 1 out of 3 women will be diagnosed to cancer. But despite of those huge number, most individuals don't know what that really means. The simplest level of cancer or cancer cells are cells that lost the ability to follow the normal control that the body exerts in all cells. In our body, we have billions and billions of cells and different function. It is very complicated process and incredibly phenomenal control, and if something goes wrong and that control is lost and particular cells escape from normal mechanism and they continue to grow and spread, that is what we call cancer. Next, let's give the definition and explanation about the two types of genetic mutation, the acquired mutation and germline mutation. First, let's discuss the acquired mutation. Acquired mutation are the most common cause of cancer. They occur from damage to genes in particular cell during a person's life. Acquired mutations are not found in every cells in the body, and they are not passed from parent to child. A genetic change that occur in a single cells after the conception of individual, the change then pass along to all cells descendant from that cell. Acquired mutation are involved in the development of cancer. So they are the examples of acquired mutation. The breast cells or colon cells. Factors that causes this mutation include tobacco, ultraviolet radiation, viruses, and the age. Next is the germline mutation. These are less common. A germline mutation occurs in sperm cells or egg cell. It passes directly from parent to child at the time of conception. It accounts for about 5% to 20% of all cancers. A germline mutation are relatively change in a DNA to affirm sex cells destined to be accordance of the sperm and zygote that conceive a single cell stage. When transmitted to a child, the germline mutation is incorporated in every cells of their body. Germline mutation play a role in genetic diseases. They play a role to in a certain types of cancer has, for example, the eyes, blastoma, womb tumor, and childhood malignant. Next would be the three types of cancer genes. The tumor suppressor genes, oncogenes, and DNA repair genes. Let us discuss first the tumor suppressor genes. Tumor suppressor genes are the protective genes. Normally, they limit cells growth by monitoring how quickly cells divide into new cells, repairing mismatched DNA, and controlling when a cell dies. These three is the important tumor suppressor, the p53, retinoblastoma gene or chromosomes 13, and the p18. Tumor suppressor gene is totally opposite of oncogenes. The tumor suppressor genes are genes that suppress the development of tumor in the dish of a people, and when the tumor loses them, it loses the ability to suppress the tumor. The very good example is the p53 tumor suppressor, a gene, and when it lasts to grow out of control. The other important tumor suppressor is the retinoblastoma gene or the chromosome 13, and the third one is the p18. Next is the oncogenes. Oncogenes turns a healthy cells into a cancerous cell. Mutation in these genes are not known to be inherited. Oncogenes are genes that is oncogenic that cause cancer. One of the most famous oncogenes is MYC. It turns out to be a very important oncogene in multiple myeloma. It is one that is so serious about the progression from benign disease to multiple myeloma. cancer. Important oncogenes in myeloma are cyclin D1, cyclin D2, cyclin D3, MAF, MAF-A, and MAF-B, FGF receptor 3, MMSET, MEK, NRAS, and KRAS. These genes are located at the chromosome's translocation. Oncogenes can transform normal cells from cancerous. And the last one is the DNA repair genes. This fix mistakes made when DNA is copied, and many of them functions as tumor suppressor genes. DNA repair genes are genes that are important in keeping genes stable and avoiding accumulation in location over time, and either tumor suppressor genes or oncogenes that they are two tumor susceptibility genes that if you activate one of those genes you're most susceptible to develop tumor and the tumor will progress faster. And that's all for my video presentation about the genetic basis of cancer. Once again, good morning and thank you for listening to my discussion, ma'am. Goodbye, ma'am, and thank you.